I would like to welcome you to Part 2 of Lecture 2 on the topic of root structure and function. This lecture is the second lecture of the plant physiology subject, which is offered as a component in our Bachelor of Agriculture and Technology at North Melbourne Institute of TAFE. Please visit our website for further information on this subject and other courses that we offer at www.nmit.edu.au. My name is Dr. Nikki Cooley, and today, like the other lectures, we will be taking a journey into the roots. Before you watch this lecture, however, please ensure that you have not only watched Lecture 1 and completed all the associated reading, Moodle quiz and DIY pracs, but you have also watched Lecture 2 Part 1. Part 1 of this topic introduces the basics on root structure and function, and that knowledge will inform you and help you with Part 2. Please note there is an associated DIY practical plant structure, form and function roots, and an essential reading list that is associated with Part 1 that I recommend you complete before watching this lecture. In this lecture we will be looking at roots and water uptake, specifically at the role of water in plants and why it's so important, root selective water uptake, cell root structure with respect to water uptake. We will examine some of the physical properties of water that are important in its uptake, the process of root water uptake, and the 3 root transport pathways. So why is water so important to the plant? The answer is not one function, but that water plays a critical role in many plant functions. Hydration of the plant and its cells are perhaps the most obvious role. Without adequate hydration, cell turgidity, which is a measure of cell hydration, will be compromised. Water is absorbed and transported throughout the plant, and when in adequate supply, can and should hydrate every cell and plant organ. Water also plays a critical role in the process called photosynthesis. We will be learning about photosynthesis in more detail in this subject. Photosynthesis is the function of converting the sun's energy into useful energy for the plant. This captured energy is fixed as chemical energy, which is then in turn fixed into sugars. As part of the process of fixing chemical energy, water molecules are split, and this is the functional role they play in photosynthesis. It is when you get slight imbalances of water that you can cause malfunctioning or less than optimal growth. So, to summarize, balancing water, its uptake, its transport, and its loss is a major challenge for all plants. The figures on the slide show us the importance of water to plants. The figure on the left-hand side illustrates a linear relationship between crop yield and crop water use. It demonstrates that the more water that is used by a plant, the higher resulting yield is produced. This association assumes that no other resources, like nutrients, are limiting. This relationship on the graph is demonstrated for both barley and wheat plants. The graph on the right-hand side of the slide shows the relationship between crop productivity, which is expressed as dry weight, with rainfall. Again, the graph shows the more rainfall, the higher the dry matter productivity. This evidence demonstrates why water is so important in agricultural productivity and in natural ecosystems. And in fact, it is the most abundant and yet the most limiting resource that plants have to manage. Plants use a huge amount of water. Most of the water that a plant uses is actually lost to the atmosphere through evaporation. This occurs during the process of transpiration. Transpiration is the movement of water from the roots out through the stomata of the leaves to the atmosphere. And about 97% of all water that is utilized in the plant is lost in this process. About 2% of water is used in the process of growth, and a little less than 1% is used in photosynthesis, when the molecule is split to aid in the production of chemical energy. Water loss is an unfortunate consequence of photosynthesis on land, and we will explore this in the photosynthetic lectures and in the transpiration lectures, which will occur later on in this subject. In summary, the loss of water is due to what is called the common diffusional pathway. This is where carbon dioxide from the atmosphere diffuses in to the leaf, and while this diffusion is occurring, water is lost. The plant requires carbon dioxide for the process of photosynthesis. Therefore, the more photosynthesis you have, in most situations, the more water you lose. To give you some kind of understanding of this magnitude, for every 400 water molecules lost, about 1 molecule of CO2 is gained. This emphasizes the challenges that plants have when managing water resources. So now that you understand how important water is to a plant, you can see, quite obviously, how critical roots are in the uptake of this water. But it is a little more complicated than just roots uptaking water. It turns out they are being selective about the uptake of this water. If you imagine yourself walking in soil that is damp, not only does the damp soil contain water, it contains the physical properties of the soil and all chemical properties that is in that soil, including salts. This is why it is important that the plant takes up only compounds that it is interested in or that it can use. If it takes up compounds that are toxic to it, this will be significantly detrimental to its growth and development. Therefore, we say that the role of a root is for the selective uptake of water. So what does it mean? How does a plant actually be selective about the uptake of water? Well, this process is quite complicated, and in order for us to understand it, we first have to understand the physical processes and dynamics of water, along with being able to relate this to the root structure, as they both work in uniformity to take up water selectively. Let us start with some structure and properties of water. The major difference between an animal and a plant cell is the cell wall. It enables large internal hydrostatic pressure, which is known as turgor. This is essential for cell enlargement, stomata opening, phloem transport, membrane transport, and rigidity of non-liquefied tissues. In the next few slides, we are going to learn about the physical properties of water. Water is an amazing compound, and there have been many textbooks written about it. Water is a polar molecule that forms hydrogen bonds. You can see from the image at the bottom of this slide, it contains one oxygen molecule which has 2 hydrogen molecules. In between each water molecule there is what is called a hydrogen bond. It is the existence of this hydrogen bond that determines whether a water molecule is a liquid or is a gas. The oxygen atom is more electronegative than the hydrogen. This means it attracts the electrons for the covalent bond. A covalent bond is a very strong bond. A hydrogen bond is a weaker bond. The water molecule is called a tetrahedral. That means it has 2 positive and 2 negative poles. This creates electrostatic attractions between the molecules and the hydrogen bonds. Water molecules can form up to 4 hydrogen bonds, as shown on the image on the slide. You probably already know this, but haven't realized it, that water is an excellent solvent. We use it in many of our day-to-day lives. For example, we may make a cup of tea and add sugar, and the sugar dissolves very well in the water, the tea. Water dissolves in a wide variety of substances. This is due to its polar structure and its hydrogen bonding, which was explained on the previous slide. It is suitable for ions, sugars, and proteins. Hydrogen bonding between water and ions and polar solutes decreases the electrostatic interactions, and in turn this increases solubility. Hydrogen bonding gives water high tensile strength. That means that the maximum force per unit area that a column of water can withstand before breaking is high. The pressure is measured in a unit called pascals or megapascals, and 1 megapascals equals approximately 9.9 atmospheres. Positive hydrostatic pressure versus negative hydrostatic pressure is what we call tension. Water can withstand pressures more negative than -20 megapascals. And this can be felt if you dive very deep in an ocean, for example. Cavitation is an expansion of air that happens in water. This tends to happen if water travels under pressure. The components of hydrogen bonding and pressure are important to understand when we are thinking and learning about cell turgidity and water movement through the plant. In this case, the water movement from the root tip through to the transport vessels, the xylem and the phloem. We will explore these concepts in more detail in the transpiration lecture too. There is a basic rule called Fick's first law that describes the process of diffusion. Fick's first law states the movement of molecules by diffusion always proceeds down a gradient of free energy or chemical potential until equilibrium is reached. What that means, put simply, is that compounds will always move from a high concentration to a low concentration, unless there is a barrier or something stopping that process to occur. You can think of this as just the natural order of the universe. This type of diffusion is known as osmosis. Osmosis is the diffusion of water molecules across a selectively permeable membrane. The net movement of water molecules through a partially permeable membrane form a solution of high water potential to an area of low water potential. Well, let us describe now what water potential is. Water potential is the potential energy of water per unit of volume relative to pure water in reference conditions. Water potential quantifies the tendency of water to move from one area to another due to a process of osmosis, gravity, mechanical pressure, or the matrix effects such as capillary action, which is caused by surface tension. Water potential is considered especially useful when understanding water movement within plants and the soil. In a simple way, it can be thought of as how much pressure is required in order to remove the water from a substance. So how much pressure would you have to apply if you had a glass full of soil-water mix? Or how much pressure would you have to apply if you had a plant and with water in it and you wanted to know what the water potential was? You could apply a certain amount of pressure, which would give you an understanding of this relation. We will learn a lot more about water potential, how to measure it in plants, and the role that it plays in plant water movement in further lectures. Root hairs play an important process as they can increase the surface area of the root significantly, and this can also increase the process of osmosis. If the water potential is more negative within the plant than the surrounding cells, the nutrients and water will move from a higher solute soil concentration to a lower concentration in the plant. There are 3 physical processes involved: simple diffusion, facilitated diffusion, and active transport. In this lecture, we are only going to deal with simple diffusion, as this tends to be the process of water movement. However, in the lecture on nutrients and roots, we will be looking in more detail at facilitated diffusion and active transport, as these play important roles in the uptake of nutrients. Simple diffusion is also called passive transport. You can think of it as the spontaneous downhill movement of molecules and encompasses Fick's law. The molecules follow a concentration gradient. They passively move through the lipid bilayer, membrane, and no transport proteins are involved. The image on the slide shows clearly this process. The lipid bilayer is the cell membrane, or the barrier. Above the lipid biomembrane is a solution that is high in concentration. If you think of each one of these dots as a molecule, you can see there are many of them. They will travel or attempt to travel through the lipid bilayer or the cell membrane. Assuming that the cell membrane does not act as a barrier to the particular compound that is moving through the cell membrane, and indeed it does not act as a barrier to water, molecules will be driven through the dynamics of the Fick's law. That is, they will move naturally from a high concentration to a low concentration. You can see from the middle part of the image this process occurring. The arrow shows the direction of movement. On the right hand side shows the image when the molecules are at equilibrium. At this point, no more transport will occur. This is passive transport. So to summarize, water will move from the soil into a root hair or from cell to cell via the process of passive transport. That is, water moves along a concentration gradient. When equilibrium is reached, water movement by this mechanism will cease. So, water enters the root via the root hair structure. The structure and the architecture of the root can alter the rate of solute and water uptake. A number of physical mechanisms play a role in the water uptake process, and we will look at these briefly now. Let us start by looking at the selective component of water uptake in plants. The selective permeability of a plant cell's plasma membrane acts as the control for the movement in and out of solutes. It is selective. It will allow compounds it wishes to pass through and stop compounds it does not want to pass through. Water easily passes through a selectively permeable membrane. Specific transport proteins in these membranes are the mechanism that controls and allows the solutes that are larger than water to be taken up. We will talk about these in more detail in the next lecture. There are principally 3 physical attributes that allow selective uptake of water. Water diffuses from areas of low solute concentration, that is, described or measured as a high water potential, to areas of high solute concentration, which confusingly is measured as a low water potential. Water will move from areas of high pressure to areas of low pressure by a process known as bulk flow. And finally, as discussed in the previous slides, cell membranes are selective to solutes. So, solute concentration, the pressure of the cell and the area, and the selective permeable membranes are what enables selective uptake at the root hair epidermis. The image on the slide is a very simplistic view or overview of the process of water uptake in plants. The point I want to make specifically here is one leading from the previous points about water potential being a measure that determines how water can flow. If you'll remember, water diffuses from areas of low solute concentration to areas of high solute concentration. Two components, the osmotic potential and the hydrostatic potential, influence the water potential in cells. This may add confusion, so when learning, remember that osmotic potential plus hydrostatic potential gives you indications of the water potential. So what we say is the water potential will indicate or say how likely it is for the water to move into the root. So let us begin to look in some detail at how and what these structures actually look like. Starting in the soil environment, we have water. This moves through to the root hair via the process of bulk flow. Once the water enters the root hair, it is transported to the center of the root to an area called the stele. Here it will reach the vascular bundles and enter the xylem. This is an overview of the process, and in the following slides we will look at this in more detail. The image on the slide shows an actual root structure, and it was a photograph from a slide of a radish, which was produced by Anne Bruce. I have modified this image to illustrate the direction of water flow as bulk flow into the root hairs. There are hundreds of root hairs on any one root tip. This allows a larger volume of water to be taken up, as the area is significantly increased by so many of these root hairs. The root hairs are actually projections of the epidermis tissue. The hairs consist of a narrow walled chamber, and they function, as I said earlier, to greatly increase the surface area, and this plays a role and this determines how architecture plays a role in the amount of water that can be taken up by the plant. This is a cross-section of a root tip. I have indicated in red a root hair, which is where the water will move into the root via bulk flow. The first structure it will come across is the epidermis. This is a selectively permeable membrane. The image on the slide is from a monocot root section. It's a cross-section of this root. Other areas I'd like to draw your attention to are the cortex, which is the area between the epidermis and the endodermis. The endodermis, which contains the capsiparium strip, which is a cell wall just outside the stele. This cell wall plays a very important role and prevents the loss of solutes from the root. Inside the endodermis is the stele, where the vessel transport structures are located, and they include the xylem and the phloem. It is in the xylem that water is translocated or moved around the plant. Here we have a slightly more magnified cross-section of a root hair, a root tip. you will see the capsicum strip here, which has the very important functional role of preventing solute loss. This image is from How Roots Are Built to Perform Their Work, from the Soil and Health Organization. I again, in red, have shown the water movement route from the root hair through to the xylem vessels. As the structure of the root plays such a critical role in water uptake, I suggest you visit the following website. The link can be obtained on Moodle. On this website is a cross-section of a root. At the bottom of the image are a number of microscopes with different magnifications. They start at ×40 and go up to a ×1000. Please visit this website and explore the root at different magnifications. It would be good for you to use this as an exercise to label the following image. In summary, passive water movement in the root occurs with the three processes in place. Diffusion occurs as this is the net movement down a concentration gradient due to the random motion of individual molecules. Bulk flow is the movement of water solutes, which does include water, together due to a pressure gradient. And osmosis is the movement of water in an outer cell. So what we've learnt so far is that water that is contained in the soil environment moves through the root hair via bulk flow, and this was shown by Kramer in 1984. It then reaches the epidermis. It is at the point of the epidermis that multiple pathways of water movement through the root can occur. There is the apoplast pathway and the symplast pathway. If the soil enters the root through its epidermis, it appears that water then travels into the cytoplasm of the root cells, and this is called the symplast. That is, it crosses the plasma membrane and then passes cell to cell through the plasmodesmata. In the non-living parts of the root, called the apoplast, that is, there are spaces between the cells and in the cell walls themselves. The water has not crossed a plasma membrane. However, the inner body of the cortex, the endodermis, is impervious to water because of a band of lignified matrix, as we've introduced earlier, called the Casparian strip. Therefore, to enter the still, the apoplast water must enter the symplast of the endodermal cells. From here it can pass by the plasmodesmata into the cells of the stele. Once inside the seal, water is again free to move between cells as well as through them. In young roots, water enters directly into the xylem vessels and the tracheids. These are non-living conduits, so are part of the apoplast. Once in the xylem, the water and minerals are deposited, and water can move in these vessels up and down the plant. This slide here illustrates and summarizes the previous slides' processes of water movement. So there are three processes. One is movement out of one cell across a cell wall and into another cell, and this is called the transmembrane route. The second route is the symplast route and is shown here in blue on the image. And the third route, which is shown in pink on the slide, is the apoplast route, and this is essentially the non-living route that water can travel through. Some details that are worth noting about the apoplast route when water moves through this route. Water can move through the cellulose cell wall and the intracellular spaces. The permeable fibres of cellulose do not resist the water flow. Water cannot pass the epidermis by this route, as the capsicum strip in the epidermis cell wall is impermeable to water. Impermeable means that water cannot travel through it. The impermeable nature of the capsicum strip is due to a compound called serbin. So all water must pass the endodermis via the cytoplasm. This is a very clever process which ensures that water uptake in the root is under cellular control. The apoplast route is important when transpiration rates are high. This is because water movement is very fast when transpiration rates are high, and it requires no energy. An essential reading for this lecture and topic is the Tays and Zeiger 2010 Plant Physiology prescribed textbook. In Chapter 4, Water Balance of the Plant, there is a section on water absorption by the root. Please read this and take note of the apoplast, transmembrane and the symplast pathways that are described. Please add your notes from this text to your lecture notes here. The following video on water movement through a plant can be reviewed to examine and expand on some of the concepts we have covered in this lecture. On completing this lecture and the associated readings, quizzes and DIY practicals, the following take-home messages and concepts should be learnt. The optimal water uptake is critical as water plays multifaceted roles in plants. You should be able to describe these roles and why they are so important. You should be able to describe the physical processes that enable a root hair to take water up. These include root hairs transfer water by the process known as bulk flow. Water movement is dependent upon both solute concentration gradients and pressure gradients. Both have to be ideal to enable bulk flow to occur. The selectivity of water entry is guided by the selectively permeable membranes. The Casparian strip is a strip that contains suberin within the root. It is impermeable to water and stops the root losing valuable solutes once they've been taken up. Once water enters the root hair, there are 3 different routes that it may travel: the apoplast, transmembrane and symplast pathways. You should understand and be able to describe these pathways. And finally, water uptake is dependent on the root structure. Understand the process that root structure and function play in this essential role. This brings us to the end of Lecture 2, Part 2.